Okay, in DNA replication. DNA replication is where a copy of DNA is created. So it's basically copying DNA to another section of DNA. Okay, DNA is made up of nucleotides, and we will represent those nucleotides by letters. So for example, if a DNA sequence A, T, C, G was to make a copy of itself, it would use its base pairing rules. A bonds with T, T bonds with an A, C bonds with a G, G bonds with a C. Okay, and using these DNA complementary rules, it's possible to make a whole new copy of DNA. So I'll show you that here. So imagine we have a section of DNA. So we have A, T, C, C, G, C, A, T. Okay, and we have another section, which is C goes to G, goes to G, C, G, T, A. Okay, and so what happened, these guys are held together by hydrogen bonds. Those hydrogen bonds are weak, but they can be separated easily, and then they'll just pop back together again. Okay, and so what happens, right, the cell needs to make an enzyme, okay, called DNA polymerase. DNA polymerase, I'll draw here in red. Okay, so DNA polymerase will come through, and it will split these hydrogen bonds. Okay, and so you can imagine this DNA polymerase is going to go through and it's going to separate these into two new strands. So I'll draw those up to the side here. So there'll be a strand of A, C, C, G, C, A, T, and another strand of T, G, G, C, G, T. No, made a mistake. Oops. See, it's easy to make mistakes. Make a mistake, you just go back and fix it. So we have 1, 2, 3, 4, so another G, T, and an A. Okay, and so what happens, that DNA polymerase, when it goes through and it's splitting those two up, okay, another one is going to come through and it's going to add those new base pairs to it. So it's going to add a T to that side, and it's going to be going the opposite way on another side. So that's going to add an A on that side. Okay, a G on that side, and a C on this side. Okay, a G on that side, and a C on that side. Okay, a C, and a G, okay, a G, and a C, A, and a T and an A, T and an A, an A and a T. Okay, and so what's going to happen, now you've created two new strands of DNA. Okay, so we've done DNA replication, where you make one strand into two strands.